RNA-seq. Regular short how many people have worked with RNA-seq data before? And how many people is this going to be the first time working with RNA-seq data? And the first few slides are a bit introductory. I'm going to explain what RNA-seq data is. So, what is gene expression and why do we care about it? I'm going to go over a bit of the history of measuring gene expression, and then I'm going to introduce RNA sequencing. And RNA-seq design, I'm going to leave until later during the workshop. There's a few steps that take a while to run, so I'm going to save that up for after that. So first, what is gene expression? Put your hands up if you're not familiar with the central dogma. So good. Everyone knows that DNA gets into RNA, gets turned into protein. We're mostly interested in protein, but since protein is really expensive to measure in the cell, we can measure RNA as a proxy for measuring protein. So what we're doing is measuring the amount of RNA in the cell. It's messenger RNA, by the way. You probably already are familiar with that. So why do we want to measure gene expression? These are just some examples. We can find out if we have two particular groups, then we can measure the gene expression in one group, measure the gene expression in another group, and compare between the two of them. That's probably the most common experimental design method. Put your hands up if you're interested in that particular two comparison things. So, and what about something more complicated? So there's other things such as determining an individual's susceptibility to cancer. If we have a particular target, such as a oncogene, we can measure that that particular gene within a patient and see if it's expressed or not. Sometimes if a gene is missing completely, that gives us a sign that something has gone wrong with the patient. Another thing is finding something in bacteria. We can usually, if it's only if we're only interested in one target, we don't use RNA sequencing because that method is a bit more complicated. We can use more low throughput methods such as plotting. So the history of measuring gene expression. I'm going to go through low throughput techniques first. These are relatively old techniques such as Northern blotting. Northern blotting was used quite commonly maybe 20 years ago, maybe even more. It relies on gel electrophoresis. So only for each particular blob, it can only target the absence or presence of a particular protein. So along here are different tissue types, and if there is a blob, then it's expressed. If it's missing, color faded, fewer amount of expression. So the problem with this is it's slow and it's low throughput, and you need a lot of DNA. Another method is reverse transcribing the RNA into PCR. It's also called real-time PCR. This measures the amount of time that you find an increase in the vector that you're trying to replicate. And if it goes higher quickly, it has a higher concentration than those that go slower. So this is PCR-based technique. So more interestingly, the high throughput technique. Using high throughput, we can have a lot of genes that we're interested in. We usually measure everything. But this generates a lot of hypotheses, such as the human genome has about 20,000 genes. We're going to test each one. Is that highly expressed in sample A, highly expressed in sample B conditions, and so on. Before RNA-seq got popular, lots of people use microarrays to do the gene expression analysis. Could I get a show of hands how many people have actually used microarrays or are familiar with microarrays? Yeah, so it's going on from microarrays to DNA to RNA sequencing. So in microarrays, each one of these dots is a probe. The intensity of the dot is proportional to the amount of expression of that probe. So RNA sequencing uses throughput sequencing to quantify RNA. It's actually used on cDNA, so we have to transform the RNA to cDNA before we sequence. And unlike microarrays, where you have each of these probes is already predefined. So RNA sequencing, you can detect new things in your sample. So you can discover SNPs using RNA sequencing, and also splice variants and gene fusions. So the problem with RNA is that most of the RNA in your sample you first extract from your cells is that it is not messenger RNA. There's a lot of things like rRNA and tRNA, and these usually aren't what you're interested in when you're doing your experiment. So we need to either get rid of these tRNAs and RNAs, or extract out the messenger RNAs that we're interested in. And there are two main methods that are used to do this, and I'm going to talk about that later. Also, there's a difference between RNA sequencing and DNA sequencing, is that you're not including the introns in your RNA. So the introns are going to be spliced out when you map it back to your reference genome. And I'll show you an example of that in the workshop. So the steps for RNA sequencing is firstly to take the RNA to rRNA to get rid of the ribosomal stuff, or you select for polyadenylated RNA. So you're familiar with messenger RNA, which usually has the poly A tail. So there's an experimental technique that can extract out, pick out these tails, and then you have your sequence, your data. Another method is ribosomal depletion. You degrade the rRNA that you're not interested in. Next is fragmentation. Actually, I have the slides for this. So you fragment your RNA, that's transcribed into cDNA. Then ligate your sequencing vectors, size select, so you know you have to alignment and you have a particular fragment size you're interested in. Then you sequence, you either get head end or single end, depending on how much you your experiment, what you specified when you got your sequencing done. So in RNA sequencing file, you usually get several million reads. 2 million is usually enough per sample if it's a simple A versus B comparison. Reads the RNA, so they're sent, they just start from random positions in the gene where you're interested in. And the current length is about 150 base pairs, usually for sequencing now, maybe longer these days, 300. But the thing about RNA sequencing is that you don't need hugely long reads. You just need them to be long enough for them to map it back to your genome. So if you're not interested in splice sites, then single end is often fine. If you want to save money and get single end, 150 bases, 100 even, depending on your organism. So typical RNA-seq workflow goes like this. First, you start off with your raw sequence reads. These are usually in FASTQ files. How many people are familiar with the FASTQ format? I'm hoping most of you, if you've been here. If you don't have a good reference, so if you're working with a non-model organism, you're going to have to do some assembly to get a transcriptome. We're going to be working with a well-annotated genome today. We're going to be working with yeast, which is a model organism. After we map it, we're going to summarize how many reads align to each gene, and then we're going to do some testing to see if these samples have genes which are upregulated from other type of position. There's also further things we can do after we get our list of differentially expressed genes. such as enrichment tests. We don't have time to go through that today, but there's an optional extension if you want to do that. So this is quantifying the reads. We map the reads, and you can see that this gene is expressed as high as this gene, the blue gene, and comparing that with other samples, we can get an idea of what is and is not differentially expressed. So the other applications for RNA sequencing is when I'm interested in ASC group comparisons. So what is everyone else working on if they're not working on simple comparisons? Does anyone have a good example? No.
If they're not what?
Of other RNA-seq applications if you're not doing simple comparisons between groups. Anyone? I had a cancer experiment a while back. We were looking for fusion genes. So that's another example of what you can do. Differential splicing is another common example. Differential splicing, you probably want paired-end reads, and longer reads are better for that. So the most common is the simple differential expression analysis. And that's what we're going to do this workshop. It's just a simple ASC analysis with 3 replicates per condition. And we're going to measure how these transcripts are changing between these 2 conditions. So replicates are really important to RNA sequencing. Replicates, you can measure the amount of variability within the samples, and that's going to be really important when we do our statistical tests later on. I'm going to leave that until we do our Cuffdiff, which will take a while. So to get started on the tutorial, there's a link in the timetable, and I want you to open that up, and you get a document like this.
When we get to that bit, can I do half Galaxy Melbourne and half on GCC?
Yep.
All right, cool.
So what we're doing today is we're going over 3 workflows. This is what they all have in common. We have 4 sequence reads, we're going to map them, and we're going to summarize the gene count and do a statistical test. So for the first workflow is Cuffdiff. Put your hands up if you're familiar with Cuffdiff, if you've heard of it. Yeah. So Cuffdiff is probably on its way out by now. It's a bit dated, but the Cuffdiff workflow, we're going to map our reads using Tophat, and we'll get band files. Then Cuffdiff will summarize recounts and do a statistical test on one package. We get our Cuffdiff files and a list of differentially expressed genes. The second technique, the second workflow, is the RNA-seq. We're going to use our previously mapped band files from the previous workflow. This is going to be a demo for the easy one, but we're going to use HTC count to get a series of count files to see how many reads have aligned to each gene. Then we're going to use the RNA-seq to do the testing and get our results. Similar for AJAR, except we're going to have a single count matrix instead of a series of files having counts, and we get our AJAR results. And lastly, we're going to visualize our results using a program called Degust. Was it Degust? Degust?
Degust, Degust.
Okay.
Take your pick.
This is a web application where we're loading our data and we can have a play around and visualize it. So first thing you want to do is go back to your timetable page. I have a few servers here. So maybe this side of the room uses first server. This side uses the second server. If you want to just click on a link.
You can choose two.
Okay.
Well,
Number 2 on my side.
yeah. Or choose whichever one you want.
Choose a random one.
Yeah, random will work.
Okay, can you keep it for a second after? How do you do that? Yeah. Galaxy.
The URL to this was available at the start. Is it just at the top?
Tiny URL RNA-GCC16.
Okay, can you repeat it?
Is it just at the top of this?
No,
No,
if I
it's
just
not.
type it in maybe.
Mango. Okay, that one.
Yeah. tinyurl.com/RNA dash
Yes.
GCC 600.
Okay, got it. Thank you.
That's your master page.
I'm going
that
to
open.
Yeah. Thank
So
you.
it's got links to the slides. And
Yeah. That's good. Easy. I need to type that.
Okay.
Yeah,
Thank you.
that's the idea. So we're one thing that's actually
So we need to make an account there.
You need to register the account and log in.
Oh, I'm already logged in. Means I already made an account. Yes.
There's one that I got Melbourne, Galaxy.
Did you get it? You can have it and I can tell you what I got. If you go to tiny URL like this one. Oh, it disappeared. Just discounting the middle one. Tiny URL.
Where do you want me to write it?
/.com/GCC.
RNA-GCC.
That's the URL. Moving this course.
So when you're registered and logged in, I want you to go to Share Data and go to Publish Histories. And the one you want is the GCC 2016 RNA.
So the one,
Yeah.
no, the one, the bare one, the shortest one.
This one. Yeah.
Okay, wait a second. Publish histories and which one the
GCC 2016 RNA.
okay, the simplest one.
The simplest one, yeah, the shortest one. That one.
Yeah, we got it.
Someone got that?
Yeah. And then we import it.
Yep, you want to import the history, which is on the top right corner.
Yeah.
You should get, what have you got, 12 past you paths.
Yep.
Are they paired end?
Paired end.
It should be in the publish history, so it should be able to yeah.
No, tiny.
This one right here, this one. Right.
Yeah. So you're on Galaxy, he's Galaxy Zone, needs to be on
No, copy and paste it.
But so you could just say Galaxy dash anything else.
Well, let's just say we've got 30 people here, all trying to align. So what have we done?
G only two C's, not three C's.
That's me.
Doing.
And one GTF file.
I know. Well, so many people here.
So you said it's 12?
Yeah.
Oh, 12 FASTQ files and one G.
12 FASTQ files. There's one GTF file. So yeah, we've got 12 files. There's two files per sample.
Yes.
So that means it's there's two conditions. And they each triple. With us. Condition A versus condition B, we get 3 on this side and 3 on that side. And each one.
There are lots of things. Just, can you give us again the tiny URL so we can just get, I got mine, but it's Windows.
Yeah, totally, yeah.
Okay, here. Let me type it. Can I do it? That's not separate. Yeah, that's it.
Be someone that's sometimes we're obviously
Speaking with a guy for the one, a lot of people. And it should take you back to the main Galaxy. It should have all the files. Then you can go back to the instructions. Do you have the original?
Because now I can just click there.
Yes, but then you can wait for it.
No, there is a, yeah, on
Yeah.
There's 2 methods. So what are the 2 treatments? Jess is on it.
This data is actually from this data, so this is like this year. So there are 2 conditions. One is we've got batch, that's for fermentation. So I think one of these was limited in the glucose, so they went to the fermentation pathway. I don't know much about glucose metabolism for you, so you can search for it. I think it's in the tutorial document if you want to read more. The 3 replicate condition, the data is hidden, as you can see from the 4 files, 6 samples, 2 files per sample. And this is actually just chromosome 1 reads, because if we had the full data set, it will take a while to run, and this is just a tutorial. So you're familiar with FASTQ, so I can just go for this. If you're not, the first line is sequence adding, the second one is where the nucleotides are stored, and the 4th is the quality scores, and there's 1 character per base. So we're going to do alignment now, and I'm going to do mine. So if you go back to the tutorial, it's sometimes easier to have half the screen with a window for the tutorial and the other half is Galaxy.
correct interest.
So here I'm doing multiple data sets, and I'm going to select all the forward FastQC files.
Okay.
You need to click on the multiple data sets file if you want to do that. How do you multiple select in Windows?
Control.
Control. Thank you.
Oh, I do the same from since I switched to math. I get the same stuff.
Yeah.
How you can make data sets here as well.
Yeah, but I want to use Tuxedo later, which doesn't like data sets, and I don't know.
Simon's better than it.
Okay, so you got Tophat gap 3 number. What tool are you using?
This is Tophat.
Are you following the instructions?
No, I was listening to her and I missed it.
Well, it's best to just try and look. You missed something.
So just refer back to this document.
Yeah, that's what you're up to.
Okay.
Okay.
This is a little bit tricky to do this select thing. On Mac, how do you hold down option thing, the command thing, the multi-select? The Apple key?
Yeah, I've always found it easier to make data collections and then submit the data collection. Yeah,
that way you select that tick and
Yeah. If you go here, you can share screenshot. If that's what you're interested in. What? Which one?
And then these are hand and read. Yeah. More than that's like some things.
So
So you're probably all familiar with alignment, and here we have only the coding sequences, so we don't have the intro, and we're going to find where they belong in the reference.
Yeah, you're using that as a more match, which will collections, I think. Yeah, but you can provide the data collections.
So 4 more reads are only one.
Yeah, choose the 3 icons in a row.
Choose RNA-seq. Okay, you're there. No, I don't have the same. What did you do? Just click there.
So you
Oh, it's not single line, choose per and as individual data sets. And but here we need to choose someone, and I cannot there.
So there's hundreds of alignments to
I know Tophat is kind of old. That's
That's
why.
an old, I mean, now, I mean, HiSat 2 is pretty something now. Star is extremely fast, but it comes at a mild cost. It's fast for a reason, right? Nothing in mind is rare. How a lot of these tools make this sort of fast is they don't align to the genome, they align to the transcriptome. Transcriptome is much smaller, but the problem with that is that
So you have to choose multiple data sets,
you might
second.
you'll miss that point that you have annotated. And they then smart.
So in this case, instead of choosing individual basket, can I make the data sets?
Yes, you know how to do that? You know how to do that?
There's that this one.
Yeah, you can make that. You can make these data sets so you can like that.
Yes, here and then.
And then you can make up 10. By matching happens to go up the store. I think you can find it all in one.
Okay, so I can do it and then use that data set. Four more.
You can use that instead if you want. I think data collection way of simplifying
So if you choose yes, we want
What is So we'll see, yeah. So hopefully everyone's got their alignment money back here.
What the hell am I on?
Have you guys heard of Hyperactivity and or any other alignments? Tophat isn't popular these days. It's a bit slow. There's another program called Hyperactivity, which I recommend, or SAR is really popular. There's also known methods such as Tuxedo and Selfish. Selfish uses k-mark counting. Tuxedo is a pseudo alignment thing. You can Google it if you're interested in these methods. These methods are really fast, so that's the thing. If you have huge amounts of data, that's also a good thing. I think the tutorial also recommends renaming these files. Yeah, renaming them. So since we're not using the insertion or deletion files, I usually just remove them. So I'm just going to delete those from my history to clean them up. So Tophat also outputs a file called alignment summary. I'm going to view it here. So this is quite useful if you want to see how many reads have line. Here, 100% of reads have line. This is obviously test data, so it's very, very clean. And about 6% have multiple alignments. So here are my 6 span files. They're called separate hits at the end. I'm going to rename these to match the sample. So the first one is going to be batch 1.
It takes a bit longer when we send it. It's normal.
Lots of people are using the server, so it's very busy.
It takes like 4 minutes and it's still kind of sending, so it's normal. I just want to
if anyone falls behind, you can go to the shared data, go to publish histories, and just load up the workflow one, and you'll see the results in case you don't want to run it and you just want to look at the data.
Okay, I got it.
So well, that has been renamed. I'm just going to search here. I have 6 span files, 3, 4, 10, take a batch. And if I go back to my laptop screen, is anyone here familiar with RGB made by the Broad Institute? Anyone? Well, RGB is a visualization tool that we can load up BAM files. So I'm going to load up batch 1 and chem1. And I'm also going to load this splice junctions bed file, which Tophat also outputted. So along here are the chromosomes of Saccharomyces cerevisiae. I'm going to click on chromosome 1. And these are my reads along chromosome 1. This is the gene annotation track. So if I zoom in on, yeah, I can see individual genes. If I zoom in closer, these things here are reads. I can show all bases. Zoom in even closer, I can see each nucleotide.
And choose only 2.
This program is usually useful for variant calling, but it's also quite good for checking how well your sample has aligned. So I'm going to hide that now.
This one, I think it is.
So here's an example of an exon-intron boundary. As you can see, the reads have being spliced out here, and this intron is actually from this splice variant. This over here is the junction trap that Tophat has outputted. This organism doesn't really have a lot of splice junctions in chromosome 1, but in humans, you'll be able to see more complex splice patterns. The tutorial actually, the workshop actually suggests using Trackster, but I'm going to skip that for today. So if you want to, you can do it on your own time, I guess.
So can you just show everyone in the tube which part they might want to?
So this
section
is
3.
Visualizing align reads with Trackster. This is under aligning. So
did anyone get that working? Section 3. Visualize.
Because lots of people are in the room and it might take a while.
Okay. Other people are having trouble. We might skip it and you can.
Yeah.
How did you rename this output file? You just call it batch 1 because.
If you go back to the thing.
Yeah.
They recommend batch 1 as a piece of them. Right. So with IGV, this top part here is actually the coverage track. So if you hover over, you can see the number of reads that have aligned to that position. That particular page. Here we have 185 around this area. And in this one, we have about 60 or 70s. So this is the Chem 1 sample, and this is the batch 1. So that might indicate differential expression of this gene. The thing about this is this isn't normalized, this is just raw count data. So you probably have to do normalization methods, then your statistical analysis on top of that. But it's still quite obvious if a gene is massively differentially expressed, like that one. That's quite obvious. This one is higher expression than this sample. All right. So I'm going to go over Cuffdiff. Cuffdiff actually combines these two steps. It quantifies the reads and then does the test itself. I'm going to select my 3 samples, my 3 batch samples in condition 1, and I'm going to compare it to the chem. Two sessions and division. It's not very
So basically, you need overlay, right? If you find we look at transcript, we look for out sections of the DNA. And so our reading from the transcript. So when we get all line back to the DNA, there's a collapse section of what's happening is being able to go and look here and say, all right, only 20% of my reading maps, or 80% of the reading maps here, 20% that doesn't. I'm going to say some 2 4 columns map. I'm going to leave it out. Whereas what will happen should happen is that region is split from 20% map here, 80% of map here. So the gap is there. But because making a cap of that size isn't be everybody. It's very, very costly in point for a lot of people improve that. So it's not this is like this one. Right. So I really and that's good to get around that. So that's why we use that. There are other slices there like this. But if you're going to do RNA-seq, a lot of the really need to use it. It should be used for that way. Okay, does that make sense? Good.
So how do I like doesn't does it let me see? Yeah, I started this part. Okay, so I'll just do it that way.
So has ever the Cuffdiff job submitted?
I'm just going in order. I think the renaming is not clear. What does it mean? So I'm still at the renaming. I have like 20 something files. So I got the one accepted hits and just put random numbers.
You say, I'll sit on the outside.
Okay. Please.
Yeah, I'll sit on.
Okay. So I generated so I generated this, I used, and I got this, like a lot of files here.
So you just go down and type it.
Oh, so I can select them. I already renamed it, but I didn't know there is some.
And then this is
Also, I'm only on the last one, so I did it right. It just went random. I mean, not random, I went from.
Yeah, I understand. So you just change it, and that will be.
That's it, yeah. Okay, so I was doing it right.
That's not. It's for me, it's like. It's not one for a while.
Yeah. Okay, so here, and what's the next step before you go? That's 6 accepted.
So now, if you get rid of that.
Okay.
Yeah, you got 4. So this will still say what it is.
Yeah, no, that would be beautiful, makes sense. Okay, so what was the next step?
The next step is to load the Cuffdiff.
Okay, so I just search Cuffdiff.
Sorry, that's it. Which you're up to. Yeah, sure. So one will get 5. Yeah. So that's what we need to do here is let's go in there and call it. Yeah, let's call it 1, 2, 8. And then we need to select top 3.
Oh, well, that's my old stuff. And why did you
Yeah, and then on this one, type sample 2 and select the bottom.
history, right?
And I think the name of sample 2 or something.
Okay.
So now Cuffdiff, so if you put on Cuffdiff.
So that went through.
And this geometric or like
Yeah, I don't know which one you meant to use. Actually, it depends on what you want to do and the way you increase the curves. In this case, I wouldn't say you're doing.
So now I
What does it say to do in the tutorial?
this
Google say what it says to do in the tutorial. Yes.
yeah,
don't need that.
I think that is
Okay.
Yes, Cuffdiff done statistical analysis as well. Or?
Yeah, Cuffdiff does the quantification and statistical analysis.
So in batch 1.
So now you've got count files.
No, it has tracking files which has FP counts,
It's like 1, 2, and 3.
is the raw counts. So, yeah.
So you've imported the Cuffdiff history.
Yeah, they'll see.
What are we doing? What are we going to do after that? Explore them.
We're gonna, well, I'm gonna quickly go over this,
Scroll down.
I think.
What else did you have to change?
So my Cuffdiff job has finished running, and I'm going to look at the one called transcript differential expression testing. It's just going to be that. Each row around here is a separate gene. This is a locus. Since I only have data for the chromosome 1s, anything that's not chromosome 1 will be a no test because there's not enough data. Or if there's not enough reads on that gene, it won't have enough it won't have enough data to do a test. So most important is the p-value and the q-value, which is the corrected after multiple hypothesis testing. I'll talk about that a bit more later. The last column is a significance column. So this is just a binary yes, no. If the q-value is significant, which we define to be 0.05 here, this will appear as a yes. So in the next Cuffdiff section, I'm going to filter this Cuffdiff data using this column and get a list of significant genes. So the tool is called filter.
How can you increase the number of significant genes?
How can you increase the number of significant genes?
Do more replicates.
Yeah, more replicates, more data.
More biological replicates will give you more statistical power to detect differential. But you can't get more. If the organism didn't behave differently, there's nothing you can do about that.
No, but I think increasing the number of replicates will increase the power of.
People do 3, but that's actually pretty insane. Ideally, you would do 20. Like that's how you get real power. And there's been experiments where people have done 40 replicates of the thing, and they've detected genes, and then they've sort of removed replicates one at a time, and done the analysis to see what happens as you go down to 3. And the difference is staggering. Like 3 barely even touches the surface. It finds all the really obvious differences, but once you get to 20, 40 replicates, you can start to detect very subtle differences. So if you want to really detect it subtle, you have to do 40 replicates.
Yeah.
It's expensive, right? 3 replicates will only find gross, quite large changes that are potentially obvious.
Yeah. So Cuffdiff uses this measure called FPKM. And FPKM stands for fragments per kilobase. So million reads. All you need to know about this is normalized by library size and by gene length. So there's actually more normalization methods than FPKM. It's I'll go to this link. This is a pretty good explanation of what FPKM is. So usually you don't express things in raw counts. You normalize by library size and by gene length. TPM is probably more popular now. It's recommended by the authors of Cuffdiff. FPKM is slightly different, but it's just similar. So the most important thing is just a normalization between samples, so they're comparable. So you have your list of filtered Cuffdiff items. I got 53 genes that were significant. I'm going to rename this file to the suggested renaming scheme. So that's Cuffdiff done. The next workflow is going to be DE-seq. And this is going to be a demo. If you want to, you could load up the data. It's in the histories, or you could just look along here. If you want to do this yourself later on, the instructions are also hidden in this, the document. So I'm using HTC count. I'm going to select the 6 spam files. The GTF file is this one with the annotations. It's not stranded. And the ID attribute is gname. So while I'm waiting for that to run, this is the DE-seq workflow as shown before. I'm going to create one count file for each of our 6 samples, and then DE-seq2 will do the testing. The tool I'm using is HTC count to do the summary counts. It counts reads per feature as defined in our 13th file, which was called gene DTF. And this will look at whatever's defined. I think I'm using Epsilon, so it'll just count. HTC count also accounts for things that align to multiple genes. So in here, this example here, this particular region has, this particular read could actually belong to this gene A or this blue gene B. It really doesn't know where it belongs unless your data is stranded. So that's another advantage of using stranded data. HTC count, if you don't specify it is not stranded, it will just discharge this read. So that's finished running. If I view one of these files, a lot of these reads are on chromosomes that aren't chromosome 1. So we just scroll down. This one is on chromosome 1. So we get 7,000 reads aligning to this gene. And I have 6 of these files. So I'm going to... It also outputs a file called no feature, which is just some metadata information. So I'm just going to delete these no feature ones.
Yeah.
So the tool is DE-seq2. You specify a factor name. If you have multiple factors, such as male, female, you might want to specify more than one factor. So I'm just going to call this condition. This one will be batch. I'm going to select my 3 batch files. So chem, with 3 chem files. And I think that's it. So this is an example of expression matrix. You have more than one multiple genes. You have a row, each row is a different gene, and you get numeric values. Usually feed in something like this into DE-seq or HR or one of these count-based methods. It's kind of hard to just visualize how it is different just looking at these numbers. The first one would possibly be that we're going to do a statistical test to look at differential expressed genes. So this is normalization. This will all be done within the DE-seq2 tool. It's already, someone's already written steps on how to do it, but so it's normalized by library size, and Cuffdiff also normalizes by gene length. Here we're just looking for library size because we're comparing within genes, and that gene length won't matter if we're doing apples to apples comparisons. So there are a few count-based methods for doing differential testing. Limma is another one. It's similar to HR. It's written by the similar authors. I think they work at the same institution in Melbourne. HR and DE-seq2 use the same statistical model. It's called a generalized linear model. I'm not fan of HR because I'm biased because I'm from Melbourne, but Boom and Limma is also a good method. They have very similar results, as you'll see when we compare our significant gene results in the next section. So come back here, let's see. So DE-seq2 has also outputted plots. Let's have a look at them. This is a PPA plot. It visualizes our multi-dimensional data into 2 dimensions. So the first dimension captures 94% of our observed variance, and you can see it's splitting along this components one axis quite nicely. So each of these dots is one of our samples. This is probably these are chem samples and these are batch samples. This is the DE-seq2 output. This is the significance value, so we can see lots of things have been significant. This is ordered by FDR, adjusted p-value. And I'm going to filter on these genes. So I'm just getting the ones that are lower than 0.05 for p-values, and I'm going to rename it. So we have a list of significant genes from DE-seq2, and next you're going to use HR to produce a similar list of genes. using a count-based method. So if you go back to the tutorial method, section 5 is going to produce a matrix of gene counts. And then section 6, you're going to use edgeR to run it. Well, maybe I should demo it first. So there. So I'm using this tool called SAM/BAM to count matrix using HT-seq code. This is slightly different to the HT-seq one we used last time. This one only produces one file with all 6 samples combined. So I'm going to So it's finished running. I'm going to view it. And I can see batch 3 samples and chem 1, 2, 3. Even for the first gene, I can see I can pretty much tell this one is highly expressed in the chem samples and low in the batch. This is artificial data, so it's going to be obvious. Usually you won't get so many differentially expressed genes, but we've particularly chosen this chromosome. Got to run the edgeR.
So I have one question. In SAM and BAM this count matrix, and also you mentioned the edgeR. Those tools are not available in Gallery and main, right?
Yeah, not in Gallery at all. So
any plan for?
We're not in charge of that.
Okay.
If you have a that you could just email the admin ask produce and so that's what people do in Gallery models.
Okay.
Yeah, and then it's going to email produce and so. The tool I'm using is called differential count models using bioconductor packages. So bioconductor is a site that has R libraries. This tool also allows us to do DSeq and DSeq2 and limma, but we're just going to use edgeR. So. This produces a similar table. This one actually has a header line on the top, so I can talk about what they mean. The first one is the name of the gene. The second is the log fold change. So are you familiar with the concept of log fold change? Yes, no? Yeah. So if we compare the samples, it's kind of hard to, because the numbers you get are really extreme, it's much easier to take a logarithm. Usually we use log 2 because it's easier, it's more intuitive. If you see a 1, then it's the change is probably 0. 2. 2-fold change, you see a log fold change of 1. So this is just on a log scale. These are p-values, adjusted p-values, and I'm going to filter on adjusted p-value.
I just figured out how you see that. Most of them all run threads, make it run faster.
Nginx. I reckon it's just the web server potentially relying on a lot of them. Yeah. Well, usually Apache spawns what Australia says it needs, but Nginx doesn't.
So I just gave it another 2 more. It was only using 2, so it's made it 4.
Well, you can safely use it like
So filtering HR gives 55 lines. So 55 genes are significant at the 0.05 level. Campaign methods are usually based on t-tests. So for each observation, we get a single dot. And samples in red, another in red. We can kind of infer what distribution we've drawn the samples from. So that's a basic t-test. But Vroom and Limma and other methods use also a method called empirical Bayes. So since sometimes we only have 3 replicates per condition, it's kind of hard to have so much variance we observe. And it can borrow information from other genes. This is a bit experienced for this tutorial, but you can read a good paper on it. Just search Vroom Limma and you'll find it. This is called a moderated t-test because it borrows information from other genes. Something else to consider, since we're doing a lot of hypothesis testing, one test for each gene is multiple hypothesis testing. And are people here familiar with this concept? I hope so. The p-value, by definition, we have a null hypothesis. We assume that these genes are not differentially expressed. But if we can assume if none of them are differentially expressed and we set a significance point of 0.05, then this is a dice result, twin-sided dice. We roll it 20,000 times, one for each gene. And you can see that if we see a 1, we'll say that's significant. And if we just accepted these results, we get about 1,000 hits, which aren't really significant. So this is kind of scary if you think about it. And there's this good paper called, this study won an Ig Nobel Prize. Have any of you heard of the Ig Nobels?
Yeah.
Yes. So they're studies that win awards because they make you think. In the study was showing images of people in social situations, either either socially inclusive situations or socially exclusive situations. The term was asked to respond, saying how the person in the situation must be feeling. This is a dead salmon, by the way, so even before it got live salmon. And it found significant results. And of course, the salmon isn't going to respond, even dead salmon. So this is why you need multiple hypothesis testing. MRIs have a lot of hypotheses testing because each area is a separate test. If an area is particularly active, it could be just noise or it could be real data. It could be real signal. In this case, it was noise.
Yeah, so an area is a 3D pixel,
Yeah.
a voxel, and so there's a lot of voxels that's measuring, and they weren't correcting them.
Well, this study shows why it's,
The functional MRI studies are renowned for coming up with associations between parts of the brain and things, and this article was to make fun of how you can't really trust most of that stuff.
So in RNA-seq and other tools, that's why we get this, we get the adjusted p-value column. Oh, yeah. So p-value is not adjusted for multiple hypothesis testing. Adjusted p-value is. So section 7 looks at how much overlap we see between these three methods. And I've published a history where you can download or you can import the results of these three methods. So I'm just going to import the history. It's called RNA_concordance. You can either do this or just follow along here. And this is going to be using a Venn diagram tool called proportional Venn. First import is Cuffdiff. I'm giving it the column with the gene names. So that's finished running, and you get a nice Venn diagram like this. See that 45 of these genes are common. So the methods are usually very similar. The next section is going to be using a web application called Degust. If you go to section 8 and open this tab. So either you can upload the count file, which you generated with the SAM/BAM to Matrix, or you can just try the demo. It's your choice what to do. In this case, I'm just going to upload my counts file. If you want to download your counts, you will have to go back to your history. So I'm going to use this all histories. And it's got this one. So BAM to GGE count matrix .xls, and you just click the download button. I've already downloaded this earlier. I'm just going to upload it into Degust. Oh no, it's not my computer. the info column, I'm going to select contig, which is the gene names. I'm going to add 2 conditions. So the first condition will be batch, selecting the 3 batch samples. Chem. Save changes and view. So each of these dots is a separate gene. You can hover over to see the gene name, like this, or you can select a gene here and it will pop up on the interactive plot. This is a heat map. So blue means underexpressed in this and higher in the chem. And if it's low, it's closer to a 0 log change. So there's not much change happening in these genes. Parallel coordinates. This just shows relative to batch. So batch is 0. This gene goes in high in chem. This is more interesting if you have multiple conditions, such as in the demo case, which I'll show you in a separate tab. The demo. So here we have 4 groups. And we can see that this particular gene is higher in GPX but goes back down in LUS. So that's pretty interesting to see if you have multiple conditions. And you can scroll down to view your particular gene of interest. So usually we do visualization before DE, differential expression testing. It's really nice to look at if, just to see if anything has gone wrong with your data. I'm going to let you play around with RNA-seq for a while. So, okay.
Yeah.
So we tried to put this in the article a couple of years ago. I know it's the visualization stage, but it will only accept Cuffdiff. Sorry. Yeah, because it knows it supports Cuffdiff. You don't have to use account. Actually, how you build it in the article.
Okay. East.
So if you're familiar with NA+, then you don't really need to listen to this. This plot is called an NA plot. Along the x-axis is the average expression. So genes here are on average expressed higher than genes here. This column is the log fold change. So anything that's higher or anything that's lower is more differentially expressed. So in this sample, I see this one, just highlight it here. You can see that in the chem sample, the log fold change is 6.68. So that's pretty high. It's a big difference. this is more likely of what you would see in a differential expression. A lot of things in the middle, these things in red. Yeah. You can also filter the output by FDR. So these ones that in red are significant, and just hiding the non-significant ones. Whatever your cutoff is, you're interested in, usually 0.15. Log fold change, if you want to set up log fold change of 1, and it's easier just to look at your genes of interest.
No, it's very well documented. I can just watch
a lot of information.
But I mean, this is also normal. I mean, what you're showing us is kind of don't have to read it down. You see
The
these.
basic too, that's good.
How do you save this? Yeah. There is a button.
Maybe master. Where are you?
This is Firefox. I want to see it. This is fine.
You can put the mouse up here, is there any? Go click back. How did you get to it?
I actually done it. I like it actually here.
So
Just do right mouse button on it.
I'm like,
one of the
That's a,
the link.
so machine on one now. This one.
It's weird,
Right.
no? How do you do it in your?
So
It's for rich people.
that my Mac costs less than yours.
so this is
That's
a
not a
simple
real
5-step.
Well,
We should
That was $1,000.
this is just a simple 5-step.
no, it was almost the same, but it was many years ago, yeah.
Basically fixing
So, link as,
data. So this is agenda and this is page
Here it says
collection. So we prepared
It would be like a CPT or
so
PDF.
forward anniversary, forward anniversary, forward
the
anniversary.
slides are HTML page, published HTML.
How do you save it?
Oh, yours are Google.
Google
This
Docs.
thing called
So you save as PDF or
it takes
the presentation.
GV.
When you click on the link, how do you save?
And then pulls out what it thinks the variance.
On Windows.
Does the basic.
Windows.
No, they have to be but it's a
It's on your browser.
It's a file. I have both Chrome and Firefox.
Well, no, it's loading the preview.
So if you go here,
I
you
can
can
take
download.
Let's do a thing
Okay.
called City Core, which is a,
No, I can't save it.
it looks for common streams
I don't know, how do you say it?
That's exactly.
and then pulls out custom and
I tried it, I couldn't.
and then you send that to.
It used to be up the top. I think.
Yeah, I don't know. I've done it before.
Given this a.
Have you got view only on it?
I don't think so.
Galaxy is smart enough to know how to run some of these things. And so it parallelizes
But the
the
tutorial was really slow and
right, okay, get much quicker. Yeah, and then it pulls all that in to go into City Core, it merges it all in together in City Core. So, yeah, right, it does it all by itself, you don't get it. And so all you need to do is say, yes, this connects it.
We have it on Galaxy.
Here's the data, and this one, so it's the faster.
Where are we?
I'll go to,
The next guy, the branch. What city? do you know?
I don't know exactly what. And it's France. It's
variant calling. I'm sure there's people who share workflows.
yeah, probably.
So there's no way to save it.
Around people don't read.
How long are you going to slide there?
Yeah,
It's tricky.
Here it is.
right, so it's like this. Yeah, these are forward movement 312 from 3 different E. coli's and here's an E. coli.
You saw it?
Yeah, I don't know how to say it.
I know one Italian person.
I don't know.
How long are you going to keep the slides there?
I won't touch it in ages.
Yeah, really? So they stay there, okay, because I can figure it out, it's fine. I got a bit lost, but the tutorial is very well documented, so I can do it anytime. My brain, I need some coffee right now, but thank you so much. So, but if this stays, that's good, I'm not going to.
This page will be up until, I don't know, I don't plan to take it down, so maybe a year. It'll be safe.
It probably won't.
Yeah, I don't think it'll, yeah.
Right,
I don't think it will be. It's in Google Docs.
Yeah, just in case something goes wrong and I delete it by accident.
No, the tutorial is very nice. It's very well documented.
Oh, the tutorial is.
Yeah,
What about this one?
You make a copy.
That's forever.
Yeah, come back in.
Unless I change it, well,
Both
yeah.
of them?
And we'll make the tree in the middle.
All right.
Because I have the, cancer research place.
What about Alvarez?
Every nation, every country has and then they start querying, okay, what's new?
It's not yet.
Obviously, does it?
You gotta go on a
Is it going
bus.
to trigger it's something so easy, just check it, do this, drag
You could walk, but
it
it's a nice
into
pace before you go.
That's what I didn't see that. So
Yeah, yeah.
Especially if you're doing the same and I'll check all the
So you
time.
have.
this is exactly what I have some mathematicians now. This is exactly output.
No, it's not very far, it's just out, yeah. Right. You gotta get out, it's fine.
Yeah, my
So kids, 3 is running there, right? And they're running across.
big motor and I was driving. it.
Behind,
Yeah, you have to see that.
So I have to take it down.
So,
that's got
Digust also has this option to do MDS plots.
And saying, running my 3 sticky jobs on
Oh, wait.
it's just running. And so they'll,
Is that the end of the
No, it's, well, I still got some slides. So MDS plots,
Everyone's pretty
yeah,
exhausted, I think,
yeah.
today. I think you can get managed to would be happy.
Yeah. So these are just similar to PCA plots.
Want me to get their attention?
Yeah.
Pretty young. So what are they actually working on here?
They're just playing with structs.
So are people still playing with Digust at the moment? Or that Digust program that visualizes, or? Because I think Jess is going to finish up with some more explanation of these are kind of important things that you need to sort of assess the quality of your data. So these are probably worth paying attention for. You can always go back and do the Galaxy part later, but this is pretty important stuff.
Thanks, Dawson.
Go ahead, Jess.
Talk loud.
I have a microphone. Why do I need to talk loud?
We'll put the microphone close.
Okay, MDS scaling. This is just a mathematical technique to visualize high-dimensional data. It just simplifies things into 2 dimensions. It's quite similar to PCA plots, which you've already seen from the DE-seq2 output. So similar things should cluster together on MDS plots or PCA plots. Hopefully you see your 3 replicates clustering together. So this is a really simplification of how these things work. These 3 plots are equidistant from each other. So they're represented in 2-dimensional space like this. But if you have more dimensions, it's kind of hard to represent it in 2-dimensional space. So it just tries to put these samples the best it can on 2-dimensional space. So the distance is just a Euclidean distance, and it tries to place these optimally. This is an example of what you'll see in your RNA-seq. This particular experiment has 4 conditions, and these 2 cluster quite nicely. This one has slight overlap, but that's a bit normal. This uses 1,000 genes. You can also choose different dimensions. So the first dimension usually captures most of the variance. The second and third, it goes progressively down, and you can see that using the workshop, the Digust. So that's that one. So this the chem versus batch. Also to choose 1,000 genes, and I can set the dimensions to 2 and 3, 4. As I get longer, I expect them to not cluster as well. So dimension 1 and 2 cluster quite well. Yeah, just Digust uses root mean square. It's not really a big difference from Euclidean distance. So usually MDS plots are generated statically, but Digust has this option to view subsets of genes and play with different dimensions. That's a good advantage of using these interactive plots to play with the data. Heat maps are another useful tool. You can see the heat map if you went to MDS plot. This is an example of a heat map. This is a nice example because there's more conditions. In this one, each separate column is a separate sample. And usually we group some genes with similar expression profiles together. So you can see a huge block and know, oh, this particular group of genes are expressed in this condition. If you've ever done an experiment with mixed up samples, then you'll know that it's often a disaster. you have to resequence samples again, and they might have a different batch effect and more variance. Yeah. So a good thing to do is check you haven't got sample mix-ups. In this experiment, this particular sample, it's called REBR group. So this is actually clustering with the VIRR group. And if we look at the heat map, this particular sample is supposed to be more similar to this one, but it's showing that it is similar to these two. So these visualization methods are a really good idea to use when you want to check your data. So after RNA-seq, you get your list of genes, and what's next? It's always important to remember that your cutoff of 0.05 or whatever cutoff you use is kind of arbitrary. You could use any cutoff and you're still going to ignore a portion of your data and look at the rest. You want to validate your lists. Often if you want to get published, maybe one of the methods like RT-qPCR. And next is probably gene ontology, looking at things that have similar functions. So if you go back to the workshop document, this optional extension has a gene set enrichment analysis. This uses a website called Gorilla. This is a bit old. I think the last updated was March 2013. So if you're working with an organism that isn't a model organism, it's probably a good idea to use our updated one. But since it's yeast and I don't expect many things to change, I could choose the yeast organism, and load my list of ranked genes and process them and get something like this to see which particular functions have higher significance or lower. So I think everyone's tired for today, and I do have some slides on experimental design if anyone is interested. But I think everyone's exhausted. It's the second day of training. It's almost over.
So what are the slides on about?
Experimental design.
Yeah, that's right.
Anyone?
Well, if you want
to look at, you can look over the slides. It's just choosing how many sequence length per end. Siglet end is probably all right if you're doing A versus B comparisons. Poly A, it's probably all right if you're doing simple comparisons of rapid depletion. If you're using microRNAs or other types of RNAs, then a bit more complicated experiments, rapid depletion is all right. But poly A is cheaper, I guess. So I think we're done for today. If you have questions, feel free. I'll be around the room until the end of the tutorial.
Thanks, Dave.